Help me study for my science exams. Enzymes, Part 2. So, last time we looked at how an enzyme can affect the activation energy and reaction rate of a reaction. But today, we're going to talk about when there's already enough. When there's already enough product, when the enzyme doesn't need to make anything else, it is inhibited. Now, there are two types of inhibitors, competitive and non-competitive inhibitors. This right here is an enzyme. Right here is what's known as its active site. This is the site where the substrates will go into it. So stuff will go in here, and then they'll come out as something else. What a competitive inhibitor will do is just bind to the active site, which means that the substrates that are trying to come in are blocked. Nothing can really get through, and the enzyme won't be able to produce what it's supposed to. The thing about competitive inhibitors is that they don't always work, because if there's enough substrate, there's a ton of stuff over here, they can just outcompete the competitive inhibitor. What a non-competitive inhibitor does is it binds to what's called the allosteric site, which is basically anywhere but the active site. Just like there, for example. So it's going to bind here. And what doing that does is it's going to change the shape of the enzyme, causing it to close up completely or most of the way to where the active site is probably like here or something else, and the substrate just can't get in there, stopping it from producing what it's supposed to. And that's how enzymes don't work.